All right, let's go ahead and get started. So if anyone is not able to run the previous cell that we did before Steve's talk and get this output, please put in the chat because it might mean that you were disconnected from the VPN. And we have to go back and run some stuff. Okay, good. So in that case, we're going to go ahead and move on to the next section. We're talking about different things that we can do to data frames, which are basically matrices of data with optional column and row names. And one of the things that we often need to do in bioinformatics data or gene expression data is to filter to reduce noise. So we were just talking about how if you have a bunch of zeros, if the row is all zeros, you could remove that row to avoid having issues with downstream applications. But if there's only one zero, you probably don't want to do that. So a filtering step will help decide which parts of your data frame are non-informative because they're mostly empty, and filter those out for you. So a common filtering method is to remove rows that have all zero values. And there's a handy function called row sums, which will sum up all of the values in each row. And then that will tell you which rows have zero sums. And there are a few steps to doing this. And this is something we're going to do in the gene expression data. So the first step is to calculate this, let's calculate the sum of every row. And we'll print that output to the screen. So go ahead and run this cell where we call the row sums function and pass to it the variable that holds our data frame data. So we see, okay, sums of each row. Row 1 sums to 6. Of course, row 10, which is all zeros, sums to 0. Now we can ask R to give us only the row sums which are greater than 0. So that'll print out just to the screen whether or not, so we're kind of asking R a question here. Are the sums of each row greater than 0? And the answer that R gives us is in the form true or false. So we get trues for all rows except for row 10, which is false. And then we can use this output to actually filter the original data frame using the slicing method, which we used above. So we can say, okay, row sums of my data frame that are greater than 0. Those are all row names that are greater than 0 that are true. Now let's use brackets to slice the original my data frame only on the rows that had sums greater than 0. And remember, we have to add this comma to tell R that we are subsetting on rows, not columns. So let's go ahead and run that cell, which since we're now subsetting using slicing brackets, we get out a data frame. And as you can see, since we sliced only to rows where the sum is greater than 0, we're returned a data frame missing row 10 because the row sum there was not greater than 0. That was false. Okay, now we have a challenge. Can you use all of this information to remove all the rows in my data frame whose sum is less than 20? So I'll give you guys a minute or two to think about that. And then when I say go ahead and put the code that you come up with in the chat and we can talk about it. Okay, I see a bunch of code here, so let's talk about this. Okay, let's see, I see a few different things here. So let's think through this, okay? So if we want to remove all the rows whose sum is less than 20, that means we want to keep all the rows whose sum is greater than 20, which is kind of like what we did here. We want to keep all the rows whose sum is greater than 0. So if I want to modify this code to only keep rows whose sum is greater than 20, then it's the same kind of logic here. You would just, instead of 0, you would have 20 here. So let's go ahead and see what that gives us. And indeed, we've removed these rows where the sums are less than 20. I see that a couple of people had the less than caret here, which is a common thing to think, but hopefully the logic that I've just walked through, it makes sense. Let me know if anybody's lost. I do notice that Hannah Lee, I hope I've pronounced your name correctly. Great. Has a greater than or equal to 20, which indeed would remove all rows whose sum is less than 20, but keep any rows that happen to be exactly 20. So very clever. Let's see if that changes the output any. In this particular case, it doesn't, but good to be exact. All right, any questions about filtering based on row sums? Okay, perfect. All right, another thing that we can do to data frames is to add, we can add a column. Sometimes you have to add a column with new data or new metadata about the samples or something like that. So that's relatively easy to do. Though, in order to add a column to a data frame, we again are going to use the bracket operator, but this time we're using it to assign a name to a new column and tell R that that new column belongs to the original data frame variable that you're working with. And the way that we then turn this whole thing, the new column, into a variable using the arrow operator. And then we can just go ahead and assign a list of values to that variable. And we have to make sure that the list of values that we're assigning, it has the same dimensions as the original data frame. So we have 10 rows in the original data frame, so I'm adding 10 values. If you try to add more or fewer, you will get an error. And in order to create a list, we are going to use a new function, which I showed you on the R manual, called C. And this simple function is just a method which takes all the arguments passed to it, which have to be comma separated, and forms a vector or list, a one-dimensional array, basically. So we can go ahead and run this cell to add a fourth column to our MyDF data frame, and then print out the revised data frame on the next line there. So go ahead and run that cell. And as you can see, we've successfully added a new column called column 4. So let's go ahead and add a new column. Call it whatever you want. You can have whatever values you want in the next cell, and then print out the revised data frame. And go ahead and put your code in the chat once you're done with that. Can you guys still hear me, by the way? Oh, good. Okay, it looks like. Oh, there we go.
Yeah, we can hear you.
Chat. Okay. The chat was frozen for me. Like I couldn't click on the enter chat message, but it's all fine now. Okay, excellent. People are putting innovative new columns in. Column 5. Very good. I'm going to do the same. Very good. All right, and when you print out your new data frame, hopefully you see your 5th column there. Let me know if for some reason you're getting an error and we can troubleshoot. Yeah, big numbers. The maximum of your data frame has now increased by orders of magnitude if you were to do summary. Okay, the next thing that is particularly useful when you're working with multiple data sets is combining data frames. So R, again, since it's designed for working with data in, well, it makes it easy to work with data in a matrix format, has a very nice function, several functions, but we're just going to talk about one now, called cbind. Now, cbind requires at least 2 arguments here, and those are the names of the 2 data frames that we're going to combine. So since we only have one data frame right now called MyDF, let's go ahead and duplicate it just for the sake of having 2 data frames. And then we're going to test out cbind. So first, let's run the following cell. We're just creating a copy of the MyDF variable in another variable called MyDF2. And let's go ahead and print out MyDF2 to make sure it looks the same. And it does on my end. And now that we have 2 data frames, let's go ahead and combine them together into one big data frame using cbind. So as you can see, we now have a data frame with 10 columns and 10 rows. And one thing to note here is that R does allow duplicate column names. But as you might recall, we have not actually assigned this output to a variable. We've just printed it to the screen. So let's go ahead and in the next cell create a variable called combined DF that holds the combined data frame. Go ahead and put that code in the chat once you're done with it. Very good. So all you're going to do here is use the error operator, or the arrow operator, to assign the output of the cbind function call to the combined DF variable. Very good. All right. And then let's just double check. What are the dimensions of the combined DF data frame? We can use the dim function and pass combined DF to that function. And as we saw up here, the dimensions are 10 by 10. So now let's think a little bit about what cbind actually did. So did cbind merge these data frames on the row dimension or the column dimension? And why do you think that is? And to answer the why question, go ahead and use the help function to look at the cbind documentation. And go ahead and put your answers in the chat. All right. So hopefully everyone can see on the screen that I've run help and passed the cbind function name to it. So we can look at the help here. So Nicholas has made a good observation here that there is a similar function called rbind, and that kind of gives you a clue. Cbind combines by rows and rbind, cbind combines by columns and rbind combines by rows. So if we had wanted to kind of stack the data frames on top of each other, we could have used rbind. Although we might have run into some issues because we have duplicate row names. But there is a way to take care of that. They can R can add numbers at the end of your row names to make them unique. So cbind merged on the column dimension, and there's a similar method called rbind to combine on the row dimension. So I'm going to comment this out again since this output is kind of long by using a hashtag, and go ahead and run this cell again to get rid of that output. All right, so once we've done everything that we want to do on our data frame, the last step is to export it back out from R, because everything that we've done, all the variables that we've created, will only be saved for that session of R. If you click on the blue tab thingy on the left, it will hide output after you run it. Yes. That is a good point. Thank you, Emily. You can just double click over here on this blue bar, and that will hide the output as well. Very good. Okay, so exporting data, just like there's a read.csv function, there's also a write.csv function. Right, altered data is only stored in memory until you export it. And you want to make sure to export data in a way that's consistent with your data structure, and write it to a file type that's going to be useful to you in the future. So similar to loading in data in R, R has several built-in functions for exporting common file types, including .txt files, .csv, and .tsv files. So here, just like we read in a CSV file, we're going to write out a CSV file. The write CSV function is similar to the read CSV function in that you need to pass to it the name of the file to which you'd like to write as a string. And also, first, we need to pass to it the variable name of the data frame that we want to write out. So let's run this cell to use the write.csv function to export the combined df data frame variable out to a file called combined.df.csv. And now we can use a new function called list.files to list all the files in our current directory and make sure that we actually successfully wrote out combined.df.csv file. So does anybody not see this new file in your directory? Okay, excellent. Okay, so now go ahead and use the next few cells to export your other data frame, mydf2, as a CSV file called mydf2.csv, but this time be specific. Here we just said write out to our current working directory. Here specify you actually want to write out to your home directory, but make sure to don't just set the path to don't explicitly set the path to your home directory. Use the homedir variable that you created previously, and remember we learned about a function that can stitch together a path. go back up and find that function, and then specify your home directory as the path, write out to mydf2.csv, and then go ahead and list all the files in your home directory. Make sure that you successfully exported that file. And once you've figured out your code, hang on to it, and I will tell you when to enter it in the chat. We can all discuss. Okay, go ahead and put your code in the chat. All right, beautiful. Okay, so here's how I would think through this. I see some people have found the file.path function. It's important that when you run the file.path function, what you pass to it needs to be your path and the end of the path, which is the file you want to write to. So it's the whole path to the file that you're writing to. So what I mean by that is we can start out with our write.csv command here, but instead of writing out combined df, right, we're writing out mydf2, and we're writing it out to a file called mydf2.csv, but we want to specify an explicit path using a variable that we created previously. So let's just remember what's in this variable. So the home dir variable points to, oh, you know what? I think, yeah, I would, the SGSU VPN kicked me off, and so I need to, can you guys still hear me?
Yeah.
Okay, 1 second. I just need to fix something here. It's not, I have to restart the kernel. One second, you guys. Sorry about this. Since the kernel restarted, it lost all of my variable names, so I just had to rerun all of these cells again. Okay, so I now have my variable names back. So where were we? All right. So I wanted to just demonstrate that the home dir variable, in case we've forgotten, can points back to a string, which is the path to my home directory. So if I want to write out the mydf2, I pass it to write.csv, I pass mydf2 variable, and then comma separated, I then want to pass the path to the file I want to write out to. So that's the file name. If I want to specify that I want to write out to the path held in this variable, I can use file.path to stitch together the home.dir variable together with the name of the file that I want to write to. And just as a reminder, when you run this file.path function, it stitches together your home.dir variable and the path held in your home.dir variable and the name of the file that you pass to it. And we noted that it adds a slash here to create a complete path. So when I run this cell, it should write out the data held in the mydf2 variable to a mydf2.csv file in my home directory. So let's do list files. Since I know I'm in my home directory, I can do this and look to see if mydf2 file is here, which it is, or I believe I can do this. I can actually list the files in whatever path is held in this variable, which happens to be the same as my current working directory, but it might not always be. Is anybody lost here? Any questions about what we've just done? Okay, someone's giving me a thumbs up. Oh, that's so nice. Not being able to see your faces is a little difficult, so I appreciate the thumbs up. Okay, awesome. Okay, so in that case, we can move on to the final and most fun, in my opinion, part of our intro tutorial, visualizations. And this will get more fun when we have our own data. But let's start out with talking about just a basic built-in function from R. So this doesn't come from any fancy package or library. We don't have to import it. It's just there in the base R installation, and it's just called plot. Very descriptive. And at its most basic, the way you call this function is you pass in an X vector and a Y vector, where both X and Y are numeric vectors or lists of numbers containing the X, Y points for the plot. So they go together to tell plot where the points should be. So, for example, we can call plot using the following parameters. As X, we can pass in the values from mydf2 column 1, and as Y, we can pass in the values from mydf2 column 4. Now, you might think that we could use the slicing syntax to extract just the one column that we're interested in each case. But remember, if we do this, that produces a subset data frame. So that has a column name and it has row names, has all this stuff that plot isn't going to know what to do with. What we need instead is just a simple vector of numbers, just the values that are held in each column. So we can use a different syntax where we take advantage of the $ operator in R, which is a way of calling a column name. This is also a way of, sometimes if you have a named list, you can call the different named components of the list. So the $ is a multifunctional operator for identifying subsets of data that have a name. Now, the data has to have a name for you to get it, because you have to pass in the name here after the $. So we can, in order to get the values from mydf2 column 1, we simply say mydf2 $column 1, and that will give us those numbers as a vector. And it occurs to me that it would be nice if we could just demonstrate this before we go ahead and do it in plot. So I'm just going to show you what happens here. If you call MyDF2 $column1, just to prove, indeed, we get a vector, and this is the way that vectors are printed out in R in a Jupyter notebook. Okay, so if the column had space and name, the $ would not work, right? We would have to use the bracket approach. Yes, so there is a workaround where if you had a space, you could put, and this works in some cases, perhaps in all cases, you can put these little, I'm sure there's a technical word for these little things, but you can find them on the same key on your keyboard as the tilde. If you surround a column name that has a space in it, or any kind of name in our name of something in a data set that has a space in it with these little quotation marks, then R will treat this as a complete name and ignore the space. It's a real good reason not to have spaces ever in data that you are passing into R. And the issue is that it's really difficult to get just a vector of numbers if you use the bracket approach. So you really do want to be able to use the $ approach when you're working in R. But great question. Okay, so having said all that, go ahead and use the $ syntax to fill in what you think the x and y vectors would be in the function call below. So remember, we want x to be the values from MyDF2 column 1, and we want y to be the values from MyDF2 column 4. And remember, for plot, you just need to pass in your x vector and then your y vector separated by a comma. Very good. Lots of answers here, which are exactly what I would have done. And if you go ahead and run this cell, which I'm sure you already have, you get a plot that looks like this. So this is just the simplest plot you can get out of R. This is a scatter plot where the values from column 1 are sitting opposite the values from column 4 on the y-axis. You can see that R has assigned automatically axes titles based on just the vector syntax that you passed in. There are ways to rename these axes titles that we're not going to get into right now, but there are also ways to make the plot a little bit more appealing to look at. So let's go ahead and first we're going to look at the help menu for plot, and then the challenge is recreate the plot, but let's focus in on two different parameters, type and call, which starts for color. And the challenge will be create a plot with both points and lines colored purple. So first, let's just take a quick look at the help documentation. Yep, default scatter plot function. You pass in X and Y. There are, again, multiple arguments which we've left blank, and the default is null. Here, for example, X label and Y label, you can pass in strings to label the plot, the axes. Let's scroll down a little bit here and find first of all, type was the first argument that I wanted you to focus on. So this is you have to pass in a 1-character string, and they tell you all the different strings you can pass in here, giving the type of plot desired. So points, you could do L for a line, you could do B for both points and lines, stair steps, etc. And then Secondly, if you want it not to be black and white, you can specify some color. And here, it doesn't say this, but you again pass in a string with the name of a color. And not all colors are accepted by R, but a lot of the commonly used ones are. And there are also color libraries that have lots of different interesting colors and names. So let's go back up and hide this help menu. And go ahead and use what's in the help menu and what we've learned so far to create a plot with both points and lines colored purple. And just hang on to your code for a couple minutes, and then I'll ask you to put it in the chat and we can discuss. Okay, go ahead and put in the chat. Good. Good. I see a lot of people in here. So if we recall, the way to create a plot with both points and lines is to pass B to type. And the way to make a purple plot, we figured out, is to pass the string purple to the col argument. Hopefully everybody has a plot that looks like this. Anybody getting an error or have any questions about what we've just done? Okay, awesome. Does anybody have any questions overall on the R intro notebook? Okay, how do you export your plot, and is there a way to rename a column? Sorry if I missed that earlier. Can you plot multiple columns on the same graph? Okay, first of all, there is code for exporting your plot, which I will find. I don't know it off the top of my head. There is a way to rename a column. Let me see. Let's finish with the graphs and then go back to renaming columns. You didn't miss it. I didn't talk about it. So Eowyn, when you say especially for the graphs, what do you mean exactly?
I guess so. My question would be like, if you have a big data set, and it's internally facing, like you're the scientist on it, the titles of the columns are kind of, you can keep them however, but if you wanted to make a presentation or give a poster, for example, and you wanted to like rename them on your graph so that it looked nice, I guess, and was very clear, if there was a way to, I don't know if renaming the column, if there's a way to just do it for the graph or you would just do it overall. if that makes sense.
I see. No, that makes sense. So you mean like you want your graph to have nicer titles here?
Yeah, yeah, like aesthetically and like to be able to communicate it clearly, like to sort of spruce up our graphs a little bit.
Yeah, no, for sure. That actually doesn't require renaming the column necessarily. And there might be reasons you don't want to rename a column, especially, for example, if you have metadata that's associated with your actual genomic data and it relies on having the same, so like maybe the column names are the names of the samples, and then you have another chart that has the names of the samples and then how long they were incubated and then what the organism type is and things like that. So you need the sample names to stay the same across multiple data files. You don't want to rename the columns, but what we can do is we can set labels for the X and Y axis that are a lot prettier using the XLab and YLab arguments and passing those to the plot function. So we could do this. So X lab equals X label. You just pass a string in, and then Y equals Y label. Oh. And so now you have nice X and Y axis labels. Okay, so if we do, for some reason, want to rename a column, let's go here. Here, okay. So let's say we want to play around with MyDF, and let's say we want to reassign column names from IDF. The way that you do this is by accessing another function in R called call names. And I will talk through this in a second. Okay, so the way that you do this is you call a function called call names. You pass in the name of your data frame, and then you pass in a list of strings. And the list of strings needs to be the same dimension as the number of columns that you have. And then you can rename those column names in that manner. I believe it is similar. Why don't you try it using row names and see what happens? I generally don't ever rename row names, so the row dot names. Yeah, it is exactly the same, except you use a different function called row names. Good question. Okay, so to go back to Amanda's question about saving a plot, so it's been a long time since I tried to save a plot from a base R package. So I would need to look this up if someone would like to chime in.
I have a guess. This is memory from a long time ago, but I think plotting in R defaults onto whatever output device you've got. But if there's a special graphics file open, like a JPEG file or a PNG file, it'll plot right into that file. So I don't think you have to take a plot and then somehow capture it and put it in a file. And because it does that way, to Eowyn's question, R is really good at publication style graphics.
Yes, that's true. And there are a series of very snazzy visualization libraries in R, which are specifically designed to create publication level graphics. So thank you, Phil. Okay. If nobody has any other questions,
Hey, Lauren.
Yeah.
Just because we've now officially completed both Intro Jupyter notebooks, I think it's a good time to show everyone how to export their Jupyter notebook and save it to their local. So if you want to walk them through that for both the R Intro notebook and show them how they can open up their saved Unix Intro notebook from yesterday and also export that.
Yes. So if you just, oh, you mean as an HTML, not as a
HTML or PDF. Yeah. So how
Jupyter
they
notebook.
can save
Okay.
it to their local machine.
Right. Okay. I was thinking, how do they save the notebook file itself? But no, I see what you mean. Okay. Okay. So if you go to File and Export Notebook as and HTML, you will be prompted to save your notebook as an HTML file somewhere on your local computer. And then you can do the same thing by going back into the file navigation over here and open up the Unix Intro notebook from yesterday. Go to File, Export Notebook as, and HTML, and then save your completed Unix Intro notebook locally that way as well.
Thanks, Lauren. Is anyone having any issues exporting your Jupyter notebooks for those of you guys who want to? If you are having issues with it, go ahead and type in the chat. As you guys are doing that, so this is going to conclude our introduction to the command line, our introduction to Unix and R commands in Jupyter notebooks. And so if anyone has any remaining questions about either, now's your chance to go ahead and type your questions into the chat or unmute your mic and ask. Internal server error when you tried to export it as a PDF. Let's see if I can...
Yeah, you can't export it as a PDF. You have to export it as an HTML, unfortunately.
Yeah, but then you should be able to, once you do have it open on a browser as an HTML, you should be able to then save it as a PDF if you want to, if I'm not mistaken.
I have a question about downloading the Python notebooks. So the Jupyter notebooks. So I downloaded the R one, but then when I open it, it says R kernel not working or something like that.
Do you have R installed in your Jupyter installation?
Yes.
You have other R notebooks that you can open at the same time?
I've actually never used R in Jupyter before this workshop, so I've never tried.
Okay. It's possible that your R installation is somehow not connected to your Jupyter installation, which is pretty easy to have happen. I can walk you through how to connect those two offline. I'll send you an email with some links and instructions.
Oh, great. Thank you.
Let's see. Can you export as an IP1B file so we can use it in Google Colab? You can absolutely export it as an IP1B file if you want to, and if you're familiar with Google Colab. HTML is just a kind of a frozen version of what we worked on today. So go ahead and do both. Download in as many formats as you're able to.
And then before we transition back into lecture mode, we have a break coming up at 2:40, but I just wanted to touch base to see, because we're just coming off a lunch, if anyone needs a 5-minute bio break right now before I start lecturing again. If you do, just type yes in the chat. If not, we will move forward right now. Okay, it seems like everyone's good. No one has any final questions about intro stuff? Can I ask a question on that? Of course you can.
Sorry, I just wanted to clarify with download or saving it to be able to open it in Google Colab, because that's what I'm familiar with using previously. When I save it, it lets me save it as such, but then when I go to, I'm not sure if I'm misunderstanding, like it's not an option for export, so it's just saving it as that file enough, or?
Yes, that's enough.
Got it. Okay. Thank you
Yeah.
so much.
Yeah, no problem.
Okay, awesome. So if no one else has any questions, I am going to go ahead and share my screen. And then can everyone see my screen okay?
Yep.
I'm just preparing. Okay, let's go to the next lecture. So this next lecture is a pretty big one. So we're going to be, most of it I will get through today. We may not make it to have time to actually start the Jupyter notebooks for RNA-seq processing today. If not, that's perfectly fine. We have about 3 hours of reserved time over the last 2 days. So we're still doing okay on time. And during this lecture, there's going to be a lot of dense material in here. And so if I'm going too fast, if you guys want me to explain something in a different way, please stop me. You can unmute your mic and ask a question throughout. We can make this as interactive as you guys want it to be. I just really want to make sure you guys are able to follow along with these concepts. Hopefully some of it's review from some of your courses. But again, if not, and if you want a further explanation on anything, please don't hesitate to ask. And I'll have the chat up and I'll try to pay attention to it as much as I can. And if I miss something, Lauren, just go ahead and interrupt me. Get started. Okay. So this is officially moving into why we are all here, which is going to begin to talk about RNA sequencing and ultimately data processing, which is what we're going to be spending the rest of our time together doing. So let's first give a little background about what came before RNA sequencing. So this image might look familiar from some of Dr. Phil's lecture of Nobel Prize laureate Kary Mullis. And his discovery is the polymerase chain reaction, or I should say invention, and it's probably one of my favorites. So this is a way in which we can use really like changing temperatures to clone pieces of DNA. So how this works is you start with your DNA of interest, and then there's usually a region within that DNA that you're interested in amplifying or cloning, which is depicted in the darker shade of blue in this little cartoon. And you need to know the sequence of the region flanking what you're interested in cloning. So that's depicted here in this light blue. And then you need some stuff to put in there along with your template DNA. So you need some primers, which are just short oligonucleotides or base pairs that are going to be complementary to the regions flanking the area of interest that you want to clone. You're also going to need some dinucleotide triphosphates, so A, T, C's, and G's, in order to clone the region. And of course, you're going to need our little molecular machine polymerase, one of my favorite molecules, to be able to actually incorporate those nucleotide triphosphates as you're creating your copy of your DNA of interest. And how this works is essentially through 3 distinct phases. So the first phase is denaturation. So we heat up our template DNA so that way we could break apart the double helix and gain access to these base pairs. And then we go through into our annealing step, and that's where our primers will anneal or will bind to the areas flanking the region of interest, because again, they are complementary to that region. And then we'll put our little polymerase to action during this third phase, known as the extension phase. And this is where polymerase will start incorporating those free nucleotide triphosphates and create the new strands of DNA. And so basically at the end of this first cycle of PCR, whereas you started with just 1 copy of your template DNA, you now have 2 copies. And then you pretty much repeat these cycles over and over, depending on how much of your template that you want to clone. And at the end of it, and this will amplify so that first you started with 1, then you went to 2, and then 4, and then 8, and that'll continue on, again, based on the number of cycles. And your final PCR product will give you 2 to the nth copies of your region of interest. And so this technology, again, it was a remarkable invention, so much so that Kary Mullis was awarded the Nobel Prize. And he actually invented this back in 1983. And since then, this technology has really been kind of harnessed, utilized, and put into different contexts to be able to give us the technologies that we have today. And so one limitation of the straight PCR is that it was difficult to quantitate. So if you wanted to quantitate how much stuff you had in there, you couldn't really do it just with standard PCR. And so quantitative polymerase chain reaction, or qPCR, was born. And just because we're going to be talking about RNA and sequencing RNA, we would start, in this case, for RT-qPCR, with mRNA. And so the RT in that abbreviation stands for reverse transcription. So basically our first step before we kind of get into the amplification would be to isolate mRNA and then convert it into complementary DNA or cDNA. So in this case, cDNA would be our starting DNA template. And then similar to what I just showed for a standard PCR, we would also need our primers, we would need our dinucleotide triphosphates, and we would also need my favorite molecule, our polymerase. Felix, you have a question?
Yeah, so how do you end up with, so if you start with 1 strand of RNA, will the thing with the arrow, the thing below the arrow have 1 strand of RNA and 1 strand of DNA?
Good question. So what happens is in this reverse transcription step, you're going to give it similar things to kind of what we gave the PCR with the DNA, where, let me go back to here. So similarly, so think about this, instead of having double-stranded DNA here, we're going to start with just mRNA, which, as you mentioned, is single-stranded. But then we're also going to be adding our primers. So they're actually random primers when you do our reverse transcription, because you don't want to introduce any bias, since you want to convert all of your mRNA there into DNA so you don't miss anything. And so you're going to have your primers again, you're going to have your dinucleotide triphosphates, and then you're going to have polymerase, a special polymerase that knows how to, or a polymerase that knows how to add in T's when in place of U's. And so as this, just like during the step for DNA, you see the first step is denaturation, right? So before we can actually do anything or make a copy of anything, we actually have to split this so that way we only have a single strand, right? So that's how the primers have access here. So that's going to happen. And I know in this cartoon, I super simplified it and have, just 1 double-stranded DNA here that we split in half and make 2. In reality, you have multiple copies of that double-stranded DNA. And similarly, we're not just going to be starting with 1 mRNA strand, we're going to have multiple mRNA strands. And so it'll work in the similar way as it does in here. So you'll start with your single mRNA, and then you'll change that into cDNA as you go through your cycles. And so it'll just keep making copies, and then the 2 complementary strands will bind together. Does that make sense?
Yeah, I think I need to think about it for a bit, but yeah, thank you.
Okay. And I also have videos to, for me, sometimes watching a video is super helpful. So I have links to videos in all of my slides really to kind of help visualize these processes and what's going on. And so after we convert our RNA into cDNA, we add in the tools that we need to be able to do PCR. So again, our dinucleotide triphosphates, our primers, as well as our polymerase. In order to make this quantitative, we also add in another molecule. So in this first example here, it's a molecule called cybergreen. And cybergreen will bind to double-stranded DNA. And when it does bind to double-stranded DNA, it will actually fluoresce. So we can see a fluorescent signal. So if there is no double-stranded DNA, it won't fluoresce. So when you start now your PCR reaction, of course, your input is your double-stranded cDNA. So you will see an initial background fluorescence. And then as you go through those PCR cycles, starting with your denaturation, that will release your double-stranded DNA. And so then the cybergreen signal goes away. And then as you make your copies, you are then creating double-stranded DNA again, which will then allow cybergreen to bind, and you get a fluorescent signal. So as you make more and more copies, you have more and more double-stranded DNA present, so you increase the amount of fluorescent signal that's being given off. And then we capture those fluorescent signals with the camera to be able to quantitate it. Of course, there's some issues with cybergreen. It's non-specific, so if there's any double-stranded DNA in there, it will bind to it, so you have the issue of possibly having off-target effects and also background noise in your fluorescent signal. So another way to quantitate this PCR reaction is by using a technique called TACMAN. And in this technique, again, similarly, you'll have your forward and reverse primers. You'll also have your template of interest, and in our case, it's cDNA, and you'll have your polymerase. But instead of cybergreen, you're going to also add in this TACMAN probe. And this TACMAN probe is going to be complementary to a region of your target interest that you're amplifying. So it's going to be a region between your forward and your reverse primer. And again, complementary to your target interest. And then what the TACMAN probe does is once you denature your double-stranded DNA, not only will your forward and your reverse primer anneal, you'll also have your TACMAN probe anneal in between them. And I probably should have mentioned that, Felix, actually. It's the fact that you have a forward and a reverse primer, you're sequencing in both directions. So that's why you end up having two complementary strands, which ultimately find each other and come together and bind to make a double helix.
I think that might have been the piece I was missing.
Yeah, sorry. Important piece. So yeah, you're sequencing in both directions. So you'll have a copy in each direction, which is what allows it to ultimately become double-stranded. Sorry, getting back to this. So then we have our forward primer and our reverse primer both annealed, as well as the TACMAN probe. And again, your forward primer is annealed to one strand, and your reverse primer is annealed to the complementary strand. And then as your polymerase then hops on and starts pulling in and adding nucleotides, it will eventually make its way to the TACMAN probe. And so initially, your TACMAN probe has this fluorescent signal, but it's being quenched by a quencher. And this is totally related to the proximity of the quencher to the fluorescent label. So as long as the quencher is close enough to that fluorescence label, it'll keep that signal quenched or silent, so that way you can't see the fluorescence. But then as your polymerase is going through and adding on your nucleotides, it'll push right through your TACMAN probe, and it'll ultimately release your fluorescent signal, because then that fluorescent signal will be far enough away from the quencher that it will be able to fluoresce, and then we detect that fluorescence. So then similarly to what happens with cybergreen is that the more cycles you go through in your PCR, the brighter your signal. And then you can quantitate that with respect to your starting signal. And so, Aylin, question.
Yeah, just to clarify, so is the TACMAN used with cybergreen? Like we're showing the TACMAN the use of the probe and quenching of that signal is what reduces the background noise? Are these two separate assays?
These are two separate assays.
Got it.
So if you were to add in your cybergreen along with this, it would kind of confuse the system and actually probably enhance your fluorescent signal a lot more quickly. And you'll also still have all the caveats of the cybergreen, like the background noise in a non-specific binding. So yeah, these are two separate assays. I've used both of them multiple times. Cybergreen's a bit more cheaper, but TACMan you usually get cleaner signal because you have that increased specificity. We'll get into the pros and cons, well, really the limitations of this in the next couple of slides. Felix.
So the purpose of this is to know when to stop, or what is the purpose of doing the quantitative PCR?
Great question. So let's say we have two samples. For our purposes, we have a flight sample and we have a ground control sample. And let's say we're really interested in expression of a certain gene that we know is important in, say, proliferation. So NF-kappa B is a good one. So we want to know if NF-kappa B expression is higher in our flight samples or our ground control samples. What we do is we give it the same amount of starting mRNA, but initially we don't know what mRNA is in there, what transcripts are in there in that mRNA. And so if, let's say, our flight sample has higher NF-kappa B, then we will have more NF-kappa B in that flight sample to start with than in that ground control sample. So as these cycles move on, that fluorescent signal will become stronger in our flight samples before it becomes stronger in our ground control samples. So then we can kind of do a comparative analysis between our flight and ground control to see, okay, when did we reach a certain point of brightness in these cycles, and whichever sample reaches a brighter point sooner, that means that there was more of that gene of interest to start with. And so this might make a little more sense on this next slide. So this is essentially the output of those quantitative PCR assays. So what we end up getting is we get these curves that have essentially three phases. And so this first is called the exponential amplification phase. That's really where things are starting to pick up, kind of in our lower cycle numbers here. And then as the cycles are cranking and we're starting to crank out copies of our region of interest, what we get into is this linear amplification phase here. And then eventually we'll be making so much of our copies of our target of interest that that signal will become saturated, and then we'll hit this plateau phase here. And so what we're looking at here is each one of these curves represents an individual sample, and I believe these are technical replicates, which is why they are falling tightly over each other. And so we have technical replicates of one sample, of another sample, and of another sample. So how you analyze this is you really want to look for this linear amplification phase. So you basically are going to pick a region in this linear amplification phase, and on the y-axis here you have your fluorescence, on the x-axis here you have your cycle number. And so you pick a location where everything is in the linear phase, and now you can start to see, okay, at what cycle number does this sample reach this amount of fluorescence, right? And so this sample here is reaching this level of fluorescence sooner than this sample here, right? Earlier in the number of cycles, which must mean that this started with more of the material that you're amplifying than this sample did. And so you kind of look at the change in the number of cycles to reach the level of fluorescence that you selected based on that linear amplification range. Anyone have any other questions that make it a little clear about what the point of this is? Okay. And I should also mention that, of course, sample handling can introduce bias. And so kind of to control for that, we also usually have a set of housekeeping genes that we know doesn't change based on your experimental condition. So for any of you guys who have done research and done PCRs, beta-actin is a very common one, although has been debated recently in the literature. So you want to look for a gene that is not going to change no matter what the condition is, and then you use that to account for any sorts of technical variation due to like sample handling and stuff between your samples. And so this was really awesome. It gave us the opportunity to be able to start comparing gene expression in a semi-quantitative manner between two samples. And so we could start to see if a sample has increased gene expression or decreased gene expression relative to another, depending on the experimental condition. But it comes with its own set of limitations. So of course, we have to know the sequence of the gene that we're interested in. So we can only look for genes that we already know the sequence of. So no chance to learn about any new genes. We already mentioned this, but a potential for non-specific binding, specifically when working with cyber green. I didn't get into this too much, but it does require primer optimization. So when you're looking for regions that flank the area of interest that you want to amplify, you have to make sure that the regions you select are unique to that gene, or else you'll start amplifying other genes without knowing it. So that takes a bit of work to get used to and to optimize primers. Another caveat of the TACMan probe is, in addition to knowing regions that flank the area of interest and making sure that they're conserved for that gene all the time, and also that they're unique, you also have to find a third unique region that's in between the two flanking regions. As I mentioned briefly at the end of the last slide, this does require housekeeping genes, so genes with consistent expression, irrespective of treatment, in order to be able to properly normalize and account for different types of technical variation. Absolute quantification of the target gene, it is achievable through this method, but you have to have a standard curve. So basically you run qPCR on a sample for which you have controlled the amount of your target is there. And so you create a standard curve. So basically you know how many cycles equals what concentration of your starting material. And that could increase the likelihood for issues if the standard curve is not properly generated. I have run into that issue before in the lab. Felix.
Well, could you talk about the second bullet again, the potential for non-specific binding?
For sure. Go back to Yeah, so in the cyber green here, unlike TACMan, where it actually has to be complementary to the region of interest that you're trying to amplify, cyber green will bind to any double-stranded DNA. So remember, in addition to your target DNA, you also have primers that you're adding in. And so just as an example, those primers can bind up on each other, and that could then create, of course, double-stranded DNA, which cyber green would then interact with and create a signal, even though you didn't make a new copy, right? It's just kind of the stuff that you added in there. That also goes along the lines of being really clever about your design of your primers. But there is a possibility that part of them can overlap, and if they do, and if they form those hydrogen bonds, create double-stranded DNA, then you'll have that off-target signaling. Also, as you're cooling down your temperature for the annealing phase, there is the possibility that some region of the DNA that's in there will fold up on itself, because double-stranded DNA is not happy. It wants to be, or single-stranded DNA is not happy. It always wants to be double-stranded. So it has a possibility of folding up on itself, in which case, if it does, again, it'll make double-stranded DNA again, which will cause cyber green to bind to it, even though a new copy wasn't made. So those are just some examples of some off-target effects that you could see with the cyber green method. Does that answer your question?
Yes, thank you.
Awesome. Okay, some other limitation of this method of comparing gene expression in two different samples is that there's a limited number of samples and number of genes that can be evaluated simultaneously. I have done 384 plate wells by hand, which I don't recommend. It's not fun. But really, at that point, you're really kind of stretching the limit of how many genes you can analyze at a time and how many samples you can add to an individual plate. So it will require multiple plates, multiple of these reactions to get more and more genes analyzed. And as I mentioned before, all samples you really should be run in technical triplicates. So for every single sample that you're looking at, I would always, best practices is do them in sets of 3. So literally the same sample 3 times, just to be really confident that the signal you're getting is indeed the correct signal. And that will, of course, limit further the amount of different samples that you can add to your experiment. And then I just have another bullet here just having you guys kind of think about what some other limitations of this technology can be. So one means of getting around particularly the limitation in the number of genes that could be analyzed at a time, we got the invention of microarrays. So I just have one or two slides covering microarrays. So just as a quick overview here, again, we would start with our two samples of interest, or sets of samples, I should say, that each underwent a unique experimental condition. So we have our control sample, in our case would be our ground control samples, and then we have our experimental group, in our case would be our flight. And then similarly to what we did before, we would isolate mRNA from our samples. We would then convert that messenger RNA into cDNA, except this time we're going to label the cDNA directly with a fluorophore. And so we're going to label it with a different color for the different condition. So in this example, we're labeling our ground control with a green fluorophore, and we're labeling our flight samples with a red fluorophore. And then what you do is you have this chip here, and inside that chip you have these little dimples or little wells. And inside each little well, you have these probes. So I kind of use the term probes, oligos, and short sequences all interchangeably. They all mean the same thing. It's a short strand of A, T, Cs, and Gs. In this case, that strand of A, T, Cs, and Gs is going to be complementary to a specific gene. So maybe this well here, this oligo is complementary to gene A, and this one here is complementary to gene B, and then this one's gene C, and etc. And I know in this picture we only see one little oligo in each well, but in reality there's actually thousands of oligos in each well. And what happens is you have this cDNA, and they're attached to these fluorophores, and then you introduce those labeled cDNA from your different groups, your flight and ground control in our example. You introduce them to your chip. And so what will happen is if gene A, for instance, is expressed in your ground control sample, but not your flight sample, you'll have that cDNA hybridize with gene A, oligo here, and give off that green signal. And similarly, if gene A is expressed in your flight samples, then you would have your flight samples hybridize and give off a red signal. And if you have gene A expressed in roughly equal quantities in both your ground control and your flight samples, you'll have both of them hybridizing to the little oligos, because remember there's multiple ones here, and you'll essentially have an equal expression of both green and red signal, which comes off as yellow. And so what you're left with after you introduce your samples to your chip, which each has a bunch of different genes represented, you then see this chip is your final product, and if the gene is not expressed in either one of your samples, you will not see a fluorescent signal, as shown here. If it's present in both samples, again in our example with our ground control being labeled green and our flight being labeled red, you would see a yellow signal, as shown here in this cartoon. If it's expressed only in flight samples or more highly in flight samples, you will either get full red, or you will get more red than green being expressed or being shown on your chip. And then the opposite is true if you have a gene that's expressed more in your ground control samples than in your flight samples, you will see a green signal for that gene. And so this is what an actual chip looks like for microarray. And so as you can see here, this big bright red dot would show in our example that this gene is probably only expressed in flight samples. And then we have some of these big just green dots. So those genes are only expressed in your ground control samples. And then we have some yellow dots here, which pretty much show equal expression of that gene in both your flight and ground control samples. And then we have kind of in between that. So for here, this kind of orangish, that means it's more expressed in your flight samples than your ground control samples, even though you do still have some expression in your ground control samples. And then we also see some kind of dimmer greens, which would be expressed in both, but more so in your ground control samples. And then, of course, you have a bunch of dots that have no signal at all, and that means that that gene is not expressed in either of your samples. That is my fast and quick overview of microarrays. Anyone have any questions about microarrays before we talk about the limitations of this technology? Go ahead, Felix.
Two questions. So first one, how do you get double-stranded DNA to hybridize to stuff on the chip, do you have to denature it, or?
Absolutely. Great question. I did not mention that. You're 100% right. Yes.
Oh, so you do have to. Okay.
Yep.
And secondly, on the right, so we've talked about black, yellow, green, and red. What's orange?
So orange would be if it is more expressed in flight than ground control, but ground control, there is still some expression in ground control. It's just that the expression in flight is quite a bit more than the expression in ground control, so it's more on the red side, but since there is some ground control expression, we do get some of that green wavelength, which causes, instead of it being a red wavelength, ultimately an orange wavelength is detected.
Oh, got it. Thank you.
And then those fluorophores are measured, right? So we can quantitate the expression of light at these different wavelengths to kind of get a relative expression of the genes in each sample. Any other questions about microarrays before I start talking about limitations? Okay, good questions so far, guys. Okay, limitations of microarrays. Again, we have this issue with only annotated genes can be evaluated, so no potential for discovery of new genes here. It requires hybridization, and always, again, when we have single-stranded DNA, it does not like to be single-stranded, so it's always going to find the first thing it can to hybridize to, and so we always have this risk of non-specific binding. You also have the issue of cross-hybridization of sequences with high identity. So let's say you have two genes, even though they're distinct genes, they might have similar sequences, even though they're not exact. And so when the DNA is being hybridized to the chip, you might have gene A, for instance, be expressed in your samples, but because the sequence of gene A is very similar to gene B, so even though it's gene A, it might hybridize to the gene B location and give off a signal there. So that's will give us, again, a non-specific binding. It's difficult to detect splice isoforms. So as we know, mRNA, once it's mature, it has undergone specific splicing. So we'll show a little picture of this later on, but there's different types or different sections of RNA. So you have introns and exons and intergenic regions. And when you're going to make your mRNA, you start with pre-mRNA, which contains both introns and exons. And then you splice out or remove those introns to bring together your exons to create your mature mRNA. But some genes will have different flavors, if you will, of their gene, in which some genes will contain all of the exons for that transcript. Some genes will only contain some of the exons, but not all of the exons in that transcript. And because of that, those sequences are going to be pretty similar in the different isoforms of that transcript, which will make it difficult to tease out and detect using this method. The dynamic range is going to be limited by the scanner. And so you have to set your scanner to pick up these fluorescent signals. And so if you set it to be able to be more sensitive to high expressing genes, you might miss out on some low expressing genes. And similarly, if you set it to detect lower expressing genes or weaker signals, it might oversaturate in some of those higher expressor genes, giving you not an accurate depiction of the expression level. The low expressing genes are highly variable, again, because sometimes it's hard to pick up that dim signal. It measures relative abundance. Of course, we're comparing expression in two different samples here, so absolute quantification is difficult to do with microarrays. There could be unequal labeling efficiency of fluorescent dyes. So one fluorescent dye might have a stronger binding than another, which could cause technical biases. You'll have chip-to-chip variation. So when you're trying to repeat studies or perform larger studies with multiple chip, you have to account for slight differences in your chips. And I just beg you guys to start thinking about what could be some other limitations of this technology. Okay, so we are going to move on to RNA sequencing now. But before we do, does anyone have any questions about PCR, quantitative real-time or reverse transcription PCR, or microarrays? Thanks, Felix. That's a really good compliment. Okay, moving on to RNA sequencing and why we are all here. And you guys will notice that RNA sequencing that Illumina does and that we're going to be talking about primarily is just a remarkably elegant use of kind of all the technologies that have come before it. And so just to kind of remind you of how sequencing happened over time, I stole this slide from Dr. Heller, so this should look very familiar to most of you. And so we started off in the late 1970s using Sanger sequencing, which is kind of an archaic way of doing sequencing. I've had professors that would always tell me how easy we have it since they did Sanger sequencing, and it's not fun. I won't get into details of how that's done unless people are interested. We could talk about it during the break. But then after Sanger sequencing, this is also how the first copy of the human genome was created, was through Sanger sequencing. And of course, these are short sequences. You can only sequence small pieces of DNA at a time, so you do have to assemble them and put them all together if you want to, say, build an entire genome. Around the 1990s is when we started seeing a transition in sequencing and how sequencing is done, and that is referred to as next generation sequencing or pyrosequencing, and we're going to get into that in quite a bit of detail over the next few slides. And but again, next generation or pyrosequencing, we're still sequencing short regions, and so if you do want to build the genome, assembly is required. Although, since we're doing gene expression, we can actually take advantage of the fact that we have a lot of reference genomes. So we also have a lot of genomes that have already been assembled, especially for model organisms that we do a lot of research with, and of course humans as well. And nowadays, we have the third generation of sequencing, which is single molecule sequencing through nanopore technology. This is kind of one of the technologies that I'm super excited about because you are no longer limited all that much by the length of the sequence in which you can generate. And so we can sequence almost entire transcripts now with pretty good accuracy. And so the accuracy of these single molecule nanopores has really skyrocketed. When I first learned about them, probably about 10 years ago, the accuracy was about 50%, and kind of all the next-gen sequencers were laughing at it. And nowadays, you can get upward of 95% or higher accuracy. And so this really is the next wave of sequencing. I don't think it's fully going to replace short sequencing assays, but we're definitely going to see a lot more of this in the future. And Felix noted a picture of Wakanda here on the bottom. Dr. Heller has a bright image of what our future is going to look like. Okay, so we'll talk a little bit about what pyrosequencing is, also known as sequencing by synthesis. And I want to apologize right now. I didn't actually realize this when I originally pulled this picture. I don't know why. This is very disappointing, but it looks like it's incorporating a C as being complementary to a T. That is not true. So guys, please pretend that either this T is a G or that this C is an A. I'm really sorry about that. Pretty embarrassed by it. Anyways, this is a schematic representation of the pyrosequencing technique. So the sequencing primer in this assay is hybridized to a single strand DNA template. So again, we have our sequence of interest here. So we don't know what the sequence is, but we do know what a region of it is because we added this on artificially and in order to be able to prime it. And so we have a complementary primer here, and then we have our polymerase, of course, except in this case, this process is going to be initiated by, instead of adding all 4 dinucleotide triphosphates, we just add one at a time. And so in this case, we have cytosine being added, and then if it matches the next base in the template, then it will be incorporated. This is showing again as being incorporated, should not be, but let's pretend like that's correct. And that reaction, of course, that incorporation is catalyzed by polymerase, and it's also accompanied by the release of diphosphate, which you see depicted here. And then that diphosphate is then converted into ATP, adenosine triphosphate, by this enzyme sulfurylase. And then that ATP is then utilized to provide energy to the enzyme luciferase, which catalyzes the release of its substrate luciferin, which is not depicted here, but that will give off a light signal, which is then photographed and read by a computer. And then the enzyme Microarrays is then introduced after this process takes place to basically quench all the remaining cytosines. So basically remove any remaining cytosines, get it out of the reaction, and then the next nucleotide is introduced. And so this kind of takes place in cycles, one nucleotide at a time, over and over again. So it's time consuming, but we are able to sequence in real time. It's definitely faster than Sanger, but of course limited in the fact that we're only trying to look at one nucleotide incorporation at a time. So what several companies did and what Illumina really perfected is being able to add all 4 nucleotides at a time. And basically Illumina does this for simplicity's sake. This is how it first started, is adding each nucleotide being tagged with a different color fluorophore. And so basically you would know what base was being incorporated in as you're generating your growing sequence here based on the color of the signal that's being released. And so that essentially is pyrosequencing in a nutshell. We are going to get into very specific details of Illumina sequencing, but just in general, any questions about just this overview of pyrosequencing? Question from Kevin. I think what the picture was trying to say was that it is trying to test whether C matches T, but it doesn't, which is why it's still green and not orange. Okay, I'll give you that. Thank you, that makes me feel better. Okay. So now that we know how pyrosequencing works, at least the general concept of it, let's talk about RNA sequencing as a whole from start to finish. So how this process works is we start with 2 groups. And so in our case, we're going to have a group of samples that were flown in space and a group of samples that were kept here on Earth on the ground for ground control. And then we take those samples. In our case, it's actually going to be the soleus muscle of mice that were flown in space or kept here on the ground. We're going to isolate RNA from that tissue. And then once we've isolated RNA, we're going to convert that RNA into cDNA. And then we're also going to add some stuff to it. So we're going to add some linkers or adapters, as you see depicted here in blue and red. We're also going to add some primers to be able to sequence the region of interest. And these adapters are going to be complementary to a location on, this is called a flow cell. And so there's going to be little oligos on that flow cell that are going to be complementary to these adapters that we add onto it. So that way it can kind of latch on to the flow cell. And then of course with the primers that we add on, we'll then be able to have a polymerase sequence it. And so after we add this stuff on, we introduce it into the flow cell, and then we allow the sequencing to take place. And as I mentioned previously, this is an example of Illumina sequencing, and so we see those different colors of yellow, green, red, and blue representing our ATCs and Gs. And so we know based on what color, what sequence was, or what base was incorporated at that position in the DNA that we're trying to sequence. And then after we do our sequencing, we are left with strands of short sequences, which we then have to figure out where it came from. And so we basically take those short sequences and we map them back to a genome of, or to the reference genome. And of course, the reference genome has to be the genome from the organism in which the samples were derived. So in our case, it's going to be mice. So we're going to map our short sequences back to the mouse reference genome and do downstream analysis to identify which genes have actually been turned on and turned off in the space flight samples compared to the ground control samples. Any questions about just the overview of an RNA sequencing experiment? No, okay. Moving on, actually we're at 2:39 right now, so why don't we go ahead and take our break, and then when we come back we'll get started taking a look at RNA and different ways to prepare our RNA to be able to be sequenced on the Illumina platform. With our last minute, is there any questions? Okay, so in that case, I will let you guys all go and take your break for the next 20 minutes. So just be back here by 3 p.m.